Welcome to the 2026 CompuCell workshop. This is going to be a very short introduction to using CompuCell, and we're going to go over almost everything in this introductory module again. For many people, this may be review. For some people, it may be new. We want to start slowly so everybody can stay up to speed and follow along effectively with the class. I need to remind people that the workshop is live streamed and distributed on YouTube, and want to thank our current and past funding sources for CompuCell. So, as I mentioned, it's helpful if you have dual monitor. I understand a lot of people don't have that, but when you're going to be doing programming and trying to follow a lecture, it's helpful to have two monitors. So today we're going to start out with a very basic review. Some of this material Hayden just covered on logistics. Then we're going to talk a little bit about some example CompuCell models, and then we're going to get into running CompuCell models using Player. And then after that, we're going to build a simple simulation using Wizard in Twedit. And then I will quickly introduce some of the ways that CompuCell models are specified. So logistics, you've seen this slide just a minute ago. Hayden Fidel is the overall course organizer. Priyab Adipok, who's on the call, is one of the lecturers. Giulio Belmonte, Pedro Del Castel, myself, James Glazier, Gilberto Tomas, Jim Sluka, and Lorenzo Vischini are all going to be lecturing. Michael Getz and Joel Vannin will be available, and possibly we'll get some help from Maciek Swat, who is the lead organizer of CompuCell, who may help explaining how to do C++ extensions next week. We have a Reddit, reddit.com/r/CompuCell3D. If you find something doesn't work, please post to the Reddit. One thing that is important if you are having problems is that a simple notification like CompuCell crashed doesn't give us much to work with. So it's pretty important to save the code that you were running, and ideally take screenshots of Player or the Python console window so that we can debug with you. We're going to be using 4.9.0. There is a trial release of 4.10, but we're going to stick with 4.9 here. Workshop resources. So there's the CompuCell website. That's probably the most important one. There are the CompuCell downloads. There are the online help manuals for CompuCell. There is the Slack channel, Google Course Drive, which was shared with you earlier. There also, for the networks part of the course, are manuals for the antimony tellurium language that we're going to be using for our simple networks introduction. And there's a quick reference guide, which I encourage you to download and print out for CompuCell. There is a Slack, I believe. Hayden can put the link for the Slack in the chat. He's done it again here. That is a good way to communicate with your other classmates and with instructors. Do remember, though, that Slack is not permanent, and so both the chat for Zoom and the Slack eventually disappear. We have a couple of quick reference guides. There is a general quick reference guide to Python, which is in the student materials folder under reference materials. There's this guide to tellurium antimony for networks modeling, and there's also a quick start guide to a CompuCell, which gives you the basic commands. In my experience, ChatGPT knows CompuCell pretty well, but not completely. And so the LLMs are very helpful for coding once you know your way around the language, but they're not reliable enough to replace coding on your end so far. There is an introduction to CompuCell in the reference guides. It's a book chapter, magnetization to morphogenesis, that goes into some of the details. Pedro will cover some of that material a little bit later this morning. Almost nobody comes to this class with a background in everything you need. You don't need to be a big specialist in anything, but you need some computing, some network modeling, some basic math bio. You need some biology, a little bit of physics, and a little bit of background on how the POTS model works. And so don't be shy about acknowledging that you may have some areas that you know better than others. That's fine. One of the things we try to do in hackathon is to team people up. So perhaps somebody with a stronger biological background might work with somebody who has a stronger computational background and so on. I usually say that you need a Wikipedia level understanding of a lot of topics. Obviously, if you're going to do research on a specific biological system, you need to know more about that. For learning Python, we tend to like the Rosalind online educational system. It's free. Network modeling, the Tellurium Read the Docs is very good. Herbert has an online textbook. Systems Biology Introductory Pathway Modeling is also quite good. If you have not done mathematical biology, the Garfinkel and Shevtsov and Guo Modeling Life, the Mathematics of Biological Systems is excellent. Really covers everything in a very clear way. There's also Essential Mathematical Biology. It's good background. And actually, Wikipedia is pretty good in the area of mathematical biology. For biology, I think Gilbert, especially the older editions of developmental biology, really are inexpensive and they're easy to read. Wikipedia, again, has good concepts. For the biophysics, which you won't need quite so much directly in this class, but which is useful to have as a background, I recommend Phil Nelson, Physical Models of Living Systems, a good introduction. Phillips, Physical Biology of the Cell, is a wonderful textbook at an advanced level. That's really a graduate level textbook, but it's full of beautiful ideas and well worth reading. There isn't actually an up-to-date book on the kind of modeling we're doing here. There's a book by Shannon Preziosi. It's very good. Cellular POTS Models, Multiscale Extensions of Biological Applications. But it's from 2013, and things have changed a little bit since then. We have our papers, but really the manuals for CompuCell are going to be the things that you're going to be working with. Somebody wanted to help us write a textbook, maybe an LLM-assisted textbook. We'd love to have the help. Okay, so I now want to break, and I'll just give you some classic examples of multiscale models using CompuCell and some motivation. We're talking about multicellular organization. That could be development. How does a fertilized egg organize into an adult? It could be homeostasis. How does an organism maintain itself in the face of insults, cell death, senescence? Developmental diseases, what happens when homeostasis goes wrong, and how does it lead to pathology? Classic example of developmental disease is cancer. In the past few years, there's been a lot more emphasis on modeling infectious disease and immune response. In fact, the U.S. National Institutes of Health have just made awards of about $250 million for modeling infectious diseases, sepsis. There is the application to biomedicine bioengineering and tissue engineering. How do you design and build tissues? Somewhat to my surprise, that's an area that has not actually made as much progress in modeling as I would have expected. And then something that may not be so obvious to people here, but is quite important area of application is toxicology. If you expose tissues to chemical perturbations, what happens and why? You could think of toxicology and drug discovery in a sense as the same thing. Any chemical you introduce to an organism is going to have an effect. If it's a positive effect, you call it a drug. If it's a negative effect, you call it a toxin. All of these are things that exhibit complex interplays of physical and biochemical mechanisms. You can't really understand any of these looking inside of a single cell. You have to look at how cells interact with each other. And so computational models then become really important to take the parts lists that contemporary biology gives us and turn it into dynamic understanding of how things work. This is based on a slide from Paul Macklin, who's another multiscale modeler. He comes from a background in cancer, thinking about the things cells do as individuals: grow, divide, differentiate, die, metabolize, mutate, move, things that cells do through interactions: adhesion, mechanics, predation, contact communication, how they interact with their environment by secreting and absorbing molecular species, how they send signals, which could be chemical or force-based or electrical, how they create, follow, and remodel extracellular environment, how they respond to fluids, and then their physical constraints. Chemicals can't move faster than the diffusion limit unless you have flow, and their mechanical limitations as well. Individual cells are relatively simple for the most part. Neurons are sort of special case, podocytes and kidney are sort of special case. And yet cells are able to organize into tissues of enormously different structures and function. And so really what's interesting in our world here is going to be saying, given a cell and the control of the cell, how do you get these large-scale organizations? That's what we're going to be focusing on in class. So virtual tissues are the kind of modeling we're going to be doing. There are a lot of different terms for multiscale, multicellular modeling. The virtual tissue term was developed by the United States Environmental Protection Agency about 15 years ago. These are mechanistic, dynamic, agent-based multiscale models. We're focusing on emergent spatiotemporal phenomena resulting from interactions between agents. And those agents we'll call objects, technically. Again, these are mechanistic models, which is not to say that you can't complement these approaches with data-based and machine learning methods. We're going to typically have quantitative submodels of object behaviors and their control. And our simulations, realistically on your laptop, you could simulate hundreds to tens of thousands of cells. If you go onto a larger cluster, you might be able to simulate 100,000 cells with our methods. There are other methods that allow you to simulate more cells with less spatial detail. And our output is going to be movies. Why do you need this? And one of the real reasons here, whether it's in toxicology or drug discovery, is that a fundamental principle of engineering control is that you want to apply your control signal, make your measurements at the same length and time scale that you care about for your outputs. However, in medicine with drugs act at a molecular scale inside of individual cells. Occasionally, they'll also modulate extracellular chemistry, but typically they act inside single cells. For medicine, at least, our high-frequency measurements tend to be whole-body measurements: blood pressure, oxygen level, chemistry in the blood, and so on. The scale we actually care about is in the middle at the tissue and organ scale. That's neither the level at which we can do our control nor the level at which we can do our measurement. And that's a recipe for failure of control and instability. And we're hope that we can use computer simulations to bridge those scales to be able to predict what happens at a system scale when you apply a perturbation at the molecular scale. And similarly, what a measurement at the whole body scale means when you dig down into the organism. The example of the difficulty of extrapolating from molecule up is the classic one, which is if I give you a pile of car parts or a list of car parts, and I ask you how is the car to drive, you're not going to be able to tell me, because the driving ability of the car isn't a property of the individual parts, it's a property of the system as a whole. That's true in biology in general, and it means that a lot of classical genomics and other kinds of omics give us the components that we need. These days, omics also tells us about interactions to some extent, but it doesn't give us the dynamic information that we need to actually be able to answer meaningful biomedical questions. Biological organization is extremely dynamic. These are some examples of embryonic development. The top one is nuclear stained zebrafish. The bottom right is a chicken embryo developing in a dish. Bottom left is blood vessels developing in a zebrafish a little bit later than the top right picture. The top one here is branching morphogenesis in a mammalian lung, showing that you have changes of cell shape, changes of cell position, reorganization of cells. Now, in adults, you don't have typically as much cell movement as that in normal intact adult tissues. I want to be careful about that because, for example, immune cells are always moving around long distances in tissues. So there's always cell movement, even in an adult. And certainly after tissue damage, wound recovery after injury, you have a lot of cell movement. Again, when we're thinking about our modeling, typically we will think of a control network. These could be gene regulatory networks, metabolic networks, signaling networks, which control cell behaviors. The cells will then interact with each other and within their environment, and then they will go on to organize tissues. In this case, what we're seeing here at the top right is what's called cytogenesis, the formation of the periodic structure that's going to lead to the spinal cord in chick and also the ribs. You can see that organizing in an animal here at the top right. This is a simulation of one side of that embryo here, where we start out with cells that are initially unpatterned. There are gene oscillations here on the right. Those interact with the gradient of an extracellular signaling molecule, in this case VEGF-A. That leads to differentiation, change of cell adhesion properties, and reorganization of the tissue. Very often these models of tissues are put inside of a whole body model of transport and dosimetry, what are called PBPK models, so that if you want to ask, if I give a drug orally, for example, what is going to happen at the level of the tissue, say in the liver here. This kind of approach is really useful when cells move, change shape, or when the behavior of every individual cell could be different and important, for example, in cancer metastasis. Okay. These kinds of models have been used in basic biology for drug discovery, toxicological discovery, for therapy design. We haven't, but there are groups, for example, at Moffitt Cancer Institute or at the University of Texas that have used personalized therapies, developed personalized therapies using this kind of modeling. If you want to look at recent CompuCell papers, in the CompuCell website, there is a page called Publications, and these are papers published so far this year that we're aware of in using CompuCell. You'll see the first one is Barrett's esophagus, so a cancer paper. The second one is large language models used with CompuCell. The third one is fat deposition study. The fourth one is cell sorting kinetics, study basically the basic biology, basic biophysics of these models. Another cancer paper, muscle regeneration paper, tuberculosis, so infectious disease and immune response, modeling of planar polarity. You're going to see that next week. Scratch assays, so this is wound healing. This is another cancer paper, cortical tension, so biomechanics, and endothelial immune cell interactions. So you could see people have worked on a lot of different topics using CompuCell. And here are some pictures of simulations going back, in many cases, quite a while. This is a simulation, detailed simulation of a single three-dimensional cell crawling on a substrate. This was a simulation to the bottom left of a therapy for diabetic retinopathy using laser photoablation. Top right is a simulation of blood vessels interacting with a growing tumor. Bottom right is a simulation of blood vessels invading the retina in a process called choroidal neovascularization after age-related macular degeneration, one of the main causes of blindness in adults. This kind of modeling has also been used extensively in the context of toxicology. There's a paper now, Nicole Kleinstra, who's the first author on that paper, is now one of the high-level officials at the National Institutes of Health. Max Lung on modeling genital development in mammals. Shane Hudson modeling palate fusion. Over the years, he's taught that model as one of the breakout sessions for many years. It's a nice model to work with. In each of these cases, you'll see there's quite an elaborate model inside of each cell, talking about molecular signaling and how that controls the cell. And then that plays out in these very beautiful spatial structural models of the tissue. We've worked quite a bit on liver toxicity, actually simulated blood flow in the liver with CompuCell. That kind of work has been done a lot by other groups now, especially the group of Dirk Dresdo and his collaborators in France and in Germany are building very elaborate models of liver damage and recovery using this kind of methodology. And that can be used also, for example, to understand individual to individual variability in response to drugs. Jim Sluka did some beautiful studies of flow and dosimetry in tissues. If you have a drug or a toxicant, the concentration that different cells see is not always the same. And this is a study trying to show how the flow in the liver leads to different exposure patterns of cells. This was a simulation done by Derek Moo, who was a high school student when he did it. He's come back and given lectures on this a few times, of immune cell maturation in lymph nodes. Again, here you have a fairly complicated signaling and control model inside of individual cells, but the cells also respond to the environment by migrating in particular patterns, proliferating and dying in particular ways. That's a particularly attractive model of germinal center response. Joel Vanin and Michael Getz, who will be lecturing later in the week, have been working on eye injury, corneal eye injury now, not retinal eye injury. Here, looking at damage patterns in response to chemical insults. That was done in conjunction with Procter & Gamble Corporation, showing some industrial payoffs for this kind of approach. Models of infectious disease. TJ Seago built a beautiful model a few years ago of influenza A infection in the lung and the various immune cells and cytokines that respond to it. That was something that could actually be calibrated against a detailed time series data in rodents. So they're actually able to get quantitative temporal prediction and agreement of all of the different molecular species and cell types. And people have used this kind of approach to try to understand things like drug dosing for molecular species. A very pretty recent paper that actually started out life as a hackathon project was studying the effect of estrogen on recovery after muscle damage. In women, estrogen levels vary a lot, especially in older women. Women with lower estrogen levels tend to have more muscle injury and slower muscle repair. And this was a very beautiful and very detailed study of the molecular control mechanisms that lead to that phenotype. At some point, we'd love to be able to integrate this kind of modeling in actual biomedical situations in what are called medical digital twins. That means actually being able to predict how health states are going to change. Probably for the immediate future, the kind of modeling we're doing here is going to be too computationally expensive to use in a medical digital twin, but it can create patterns that can then be surrogated using AI methods to do fast prediction. Another place that this is showing up a lot, at least in the United States, are called new approach methodologies, NAMs. The NIH, FDA, and EMA are moving away from animal testing. And that means you are working with in vitro studies. You're going to be working with cells in a dish, not cells in a whole animal. And that means you have to do what's called in vitro to in vivo extrapolation, IVIVE. And this kind of virtual tissues work that we're doing is essential to this in vitro to in vivo extrapolation. So it's of great importance both biomedically and clinically and as a regulatory question now to use these kinds of methods. A lot of grant opportunities in the US and Europe for that. These kinds of approaches let you change the detail of resolution, so you can work as a variable power microscope. You could simulate graded perturbations. You can understand when you have amplification or suppression of feedback of perturbations due to feedback. You can back out hard to observe parameters and test hypotheses that might not be possible to do in a wet lab experiment. You don't need animals. Simulations are cheap, so you can do more combinatorics than you would be able to do in experiments. They're also quite aesthetically interesting, and so they're good for communicating and visualizing information. And they're a great way of knowledge integration. They're a great platform for knowledge integration and reuse. Building models is not easy, and it reveals what we don't know. And that sometimes is a bit intimidating, but the exercise of building a model of this kind can really be useful in terms of understanding what experiments need to be done. It's also hard. You've got lots of components in number and type, lots of parameters, which are not always easy to pin down. A lot of heterogeneity. Two cells of the same type aren't really the same. There is never complete detail. We're always phenomenological. And comparison between experiment and simulation isn't always obvious. If the simulation produces a movie, how do you compare the movies? Biology is stochastic. You never get the same thing twice. Different replicas produce different results. You can't just do averaging on that. AI methods are helping us do some of the comparisons that were hard to do before. And of course, you may be missing some basic processes or interactions. You have large range of length scales. I was talking to one of the students yesterday. The range of length and time scales in biology is quite large. Sometimes the timescales separate, which makes life easier. Sometimes they don't. And coupling can be tight. So if we're going to be representing cells computationally, we can do it in a variety of ways. There are what are called center models, which are point-like, would be like molecular dynamics models. There are very detailed finite element models of cells, which are very beautiful. There are blob models called subcellular element models. There are what are called vertex models. CompuCell uses what's called a lattice-based approach. They're all ways of representing the shapes and behaviors of cells mechanically. CompuCell itself, by now, if you've loaded it, you've seen it, consists of two components, a customized editor called Twedit++, which we're going to primarily use in this class. These days, I think people are more and more using things like Visual Studio Code for doing their primary programming. And then there's a simulation execution control environment called Player. If we were going to build CompuCell today, starting from scratch, we probably wouldn't do it this way. We'd probably integrate it into something like VS Code from the beginning. And a lot of the features that are available in Twedit are now available in VS Code as well. And Pedro will show you how to use it. The multicellular models are specified in CC3D, ML, and Python. We'll come back to that a little later. Subcellular and network models are specified in other standard languages, SPML, CellML, Antimony, MapBus, BioNetGen, and so on. You can run on AWS if you want to. Of course, in situations like that, you have to pay for your computer time. CompuCell provides a number of basic object types and properties and behaviors and interactions, chemical fields and their diffusion and decay, generalized cells, which are basically going to be cells or portions of material, which have things like volume, surface area, compressibility, can do things like move, grow, divide, or die, can stick to each other, respond to chemical fields in the extracellular environment, differentiate, secrete, and absorb. We're also going to have a finite element structure called the link that could represent a junction between cells or philopodia, or simply the mechanical properties of cells. This is effectively creating a finite element structure, which gives you more solid-like behaviors, and then networks. Networks modeling in CompuCell uses a package called libRoadrunner, which allows you to write ODE models, rate equations, and also Gillespie models. If you're interested in using Boolean network models, there's MapBus that provides that. If you're interested in rule-based models, there's BioNetGen. So that's enough background. Now I'd like to have us actually run a simulation. So at the moment, we are going to just run a simulation that's packaged with CompuCell. Okay, so people who are using Conda may find that they have to download the demos package for CompuCell separately. If you did a one-button install, the demos are built into CompuCell. If you use a Conda install or you're on Linux, sometimes you'll find that the demos weren't downloaded and you'll have to pull them down separately. If you're having a trouble, you could look at CompuCell3D.org, source bin hash install demos for how to do it. So CompuCell comes packaged with a number of small demonstration simulations. They're not perhaps the best ones. I would love to have a group of people who develop new demos for CompuCell and reorganize the demos we have. But to get some practice launching and running CompuCell, we'll just launch CompuCell and run a simulation of angiogenesis. Yeah, there you go. Again, if you did a one-button install of CompuCell, you don't need to worry about this. It's only if you're using the Conda install that you have to worry about it. So we're going to be running a simulation of angiogenesis. We're not going to get into the details now. We'll come back to this later in the week. But this was work trying to understand how vascular endothelial cells in an embryo organize to form capillaries, either when they're distributed, which is called vasculogenesis, or when they're already forming blood vessels, which is called angiogenesis. And this is work done originally by Roland Merks, who's now an institute director at the University of Leiden, who was then my postdoc from almost 20 years ago. Well, 20 years ago. The simulations are going to be implemented as CC3D ML. The Python doesn't actually do anything in the simulation. Unfortunately, when you actually look at the code, so we're not going to look at the code, the representation of the model is a little strange in the demo. But for today, since we're not going to be editing it, it won't matter. We have blood vessels. We have cells, endothelial cells, that are going to be organizing to form blood vessels. The original question was, is the mechanism of primary vasculogenesis and sprouting angiogenesis the same? And there were experiments that showed that cells in vitro, cells, endothelial cells, move up gradients of vascular endothelial growth factor A. They secrete a chemical and they respond to it themselves. There was also a set of experiments done by people that we were working with in Kansas City, looking at blood vessel growth in chick embryos, and showed that there were particular molecular perturbations that interfered with that growth. In particular, there's a molecule called VEGF-A, which suppresses chemotaxis, that is the chemical response of cells to the extracellular chemical species, at cell-cell interfaces. And that if you suppressed VEGF-A, then you didn't get sprouting angiogenesis. And so that led to a simple simulation where you have cells. These cells secrete vascular endothelial growth factor that decays in the environment. If all surfaces of the cell respond to the chemical, they aggregate to form blobs. If only the free surfaces of the cells respond to the chemical, you get vascular patterning. And this is an experiment by Davide Ambrosi showing endothelial cells in culture organizing to form vascular patterns. And so we have some basic physics here. We have cells. The cells move around randomly. VEGF, vascular endothelial growth factor, secreted into the environment and decays. The cells respond by moving up gradients of vascular endothelial growth factor. Okay, so now I want everybody to launch CompuCell. In Windows, if you go to the bottom in your search bar and you type CompuCell, you should be able to launch CompuCell that way. In Mac, you're going to have to find the CompuCell install and launch it. In Conda, you're going to have to activate your Conda environment and then launch CC3D.Player. If CompuCell is running, you should have a screen that looks like that. You may have to resize it depending on what you're going to do. And on a Mac, the presentation may look a little bit different from on a PC. Okay, I'm going to assume everyone has it running. Again, if you need help, please reach out. So this is just going to be a very simple tutorial for Player. Player has a lot of features. I'm not sure I know all of the features. Every time there's a new release, it gets fancier. So don't worry if you don't catch all of them now, but there are a few basic things that you need to know how to do. So the first thing you'll see is that there is going to be a list of a row of small icons at the top of the screen. There's 3 buttons that are round, blue. The first one is play. The second one is step. The third one is pause, and the fourth one is stop. Then there's a file handling menu, a little wrench and screwdriver that are settings, which are quite useful. Then something that will launch Twedit, the editor, a zoom menu, and then this window here, the two stacked windows, creates an additional viewing window in your screen. The first thing we're going to do is open up a demo, and it should actually automatically be able to find the demos. However, if you're not finding them because you put them somewhere else, for example, in a conda install, if you and then configuration, you'll get a window that says CC3D preferences, and then there's a setup window, and inside of that, there's a demos directory. And if you've installed that demos folder somewhere other than inside of CompuCell3D, then you'll have to point to that. If you did a one-button install, you're not going to have to do this. Okay, so now go to the File menu, select Open Demo, and then there should be a demo browser, and we should be able to find angiogenesis. Let me see if that works on my computer. Okay, I'm going to go to the File menu. I'm going to do Open Demo, and I'm going to select angiogenesis. You can also go to File, select Open Simulation, and then you should be able to find it that way as well. So go to File, Open Demo, and select angiogenesis. Yeah, here's the angiogenesis image. Alternatively, you can hit Open Simulation File here. That will open a search window, a file dialog, and you can navigate to their demos folder, which is inside of the CompuCell3D folder, and open up it has a slightly awkward path name, CompuCell3D/demos/book chapter demos angiogenesis. Now, every time you run a CompuCell simulation, there's a log created of the simulation run, and that will be in the CompuCell working directory. That's also something you can find looking at that directory here under CC3D preferences. There's workspace directory here. Nelly, good point. We haven't actually run anything, so what you should have seen when you launched, let me share the screen again here. It's going to be a bit small here, which is why I prefer to show the slides. When you open that simulation at the top, the name of the simulation should show at the very top, angiogenesis/angiogenesis.cc3d. So, as I just mentioned, every time you run a CompuCell simulation, Player logs all of that information. That's best practice because you never know what you're going to need, but it can accumulate a lot of stuff. And so it's not unreasonable to go back and delete these at any time. I would recommend not putting your working directory being your desktop or a directory that has anything other than CompuCell simulations in it. Okay, so now we are going to run CompuCell. When you're doing logging, again, you will go to that configuration menu here, Tools, Configuration menu. You could either get to it by hitting this icon here, going to Tools and Configuration, Setup, and then controlling the workspace directory. Again, if you did a one-button install, you're not going to have to change anything, but this is something that you may want to do if you're going to run actual projects. You may want to save the data from your projects at a particular location. Okay, so now let's actually run the simulation. We have the Run button, the blue button all the way to the left, the Step button, which single steps the simulation, circle, second button over, the Pause button, third button over, and Stop button, fourth button over, with a square on it. Don't hit stop unless you want to end the simulation. You can't restart. If you hit pause, you could keep going from where you were. If you hit stop, Player will actually close and relaunch. Player is designed so that when you make a change to your code, it reloads the code to the latest version and runs it. Now, some versions of CompuCell have had some issues with updating to the latest version of the simulation. And so you'll have to see how it works in 4.9. You can resize the window just by pulling on the corners as you like. Occasionally, not so much in newer versions of CompuCell, Player could get into an illegal state. There's a hidden file called _settings.sqlite. If you find that and delete it, it should fix Player. I hope nobody runs into that, but I'm letting you know in advance. If you need help with that, that's okay. Unfortunately, the toolbars themselves cannot be resized. Those are fixed by the resolution of the screen. So if they're too small, my suggestion is to reset the screen resolution to be a lower resolution. That might be a nice thing to add on to CompuCell. Again, this SQLite, I don't think you're going to run into this problem, but if you do, ask us and we'll help you out with it. Okay. Occasionally, you'll have a couple of things that can happen with Twedit and Player. If you've opened one in a multi-monitor configuration and you remove the external monitor, it may reopen where it was off your screen field of view. Sometimes the pull-down menus will pop up in unexpected places. And that could be rather frustrating, and I don't have a good answer to that, except you have to look around. They're there. The pop-up menus are there, but they're not always clear. They may not show up where you expect. In Macs, if the pull-down menus appear but are empty, that's a settings issue, and set the default theme. The light and dark themes for Mac will not work with Player. Okay, so now in your simulation, hit the little blue circle, white circle and blue circle, second from the right, which is step. And you should see a screen that looks like that. You may have to resize your window. Let's see what happens when I do it. Okay, that's what I get. Do people get that, or they get something else? You may only have one window pop up, and it may be configured differently from others. You've got one 2D window, great. Okay, if you want a second window, hit this little new graphics window icon here, the one all the way to the right in the icon bar, or you can go under Windows and say new graphics window. That'll give you a second window. If you are on Windows machine, there's this nice command here under Windows called Tile that will automatically lay out your screen for you. Unfortunately, that's not available on Macs. Okay. Why don't people try adding a second graphics window here? You can add new graphics window up here or hit this button here. You'll have two, and then you can select what you're going to display. This is cell field, and indeed it pops up on my other screen. Cell field, here VEGF, VEGF. I'll show you how to select between 2D and 3D graphics in just a second. I just want to make sure everybody has something on their screen. Is there anybody who doesn't have anything on their screen at the moment? Okay. All right. Again, if you're having trouble, please reach out to one of the instructors. Okay, so now hit play and let this run. Again, on Macs, you may have to set default theme here under setup. Okay. Within Player, within Player, there are a couple of settings that you're going to have, which you're going to use a lot. One of them is 2D versus 3D. So here, it's a little hard to see because of the layout. All the way on the left, there's a pull down, and for some reason, in my screen at the moment, it's popping up on my other screen. And I could select XY, which will give me a 2D plot, or I could select 3D, which will give me a 3D plot. Let's see if I pull this one down. Here it pulls up in the right place. 3D, XY, XZ, YZ. So select 3D. Select 3D. And if you've opened a second graphic window, make sure that one of them says cell field and the other one says VEGF. Okay. Everybody got that? And now hit play. Do people see something that looks a little bit like that? All right. Does anybody have problems? Is it not running or not able to see anything? Under visualization in Player, you can turn on a variety of rendering aspects of Player. For the most part, we'll leave it in the default. Sometimes we will not want to represent cell borders. Whenever I make a major change to the rendering, I'll have to hit run again. It'll pause the simulation. Most of the visualization controls are in that Tools configuration menu. There's cell fields and colors. There are 3D settings as well. So I'd encourage you to pull down the Tools configuration, go to Colors and 3D, and explore for a few minutes what your options are. After you've made a change, you're going to have to go back and hit run again. So for example, here, I want to make my cells green. I select Preferences, Cell Colors, and then I select the color of my cell. For the chemical field, I have to decide if I'm going to visualize the chemical field in 2D or 3D. If I do it in 3D, it'll basically show the field at the surface of the cells unless I tell it to show scalar contours. So here I've selected scalar contours show and how many isosurfaces I want. Again, don't worry. This is going just a basic introduction. You'll come back and you'll have plenty of opportunity to play with Player in more detail. I want you to get a sense of, you're going to have to do some exploring of all the features Player has. For example, the movie maker. And I want people to realize, especially this configuration window, you have a lot of options. It's worth just walking through systematically all of the different pages that are present there, the tabs that are present there, and exploring them. So are people able to get this to run? This is VEGF in 3D. Here's the VEGF field in 2D. Going back to 3D, I'm going to go to that settings menu here, field. I could turn off the scalar contours, and you see I see just the chemical field at the surface of the cells. Got to turn the scalar contours back on, and it'll give me the isosurfaces. Okay. You can pan with your mouse. You can rotate. You can zoom in and out. If you lose where you are, if you hit the letter R over the window, it'll reset it. There's a question about where no, you always have a choice of 2D versus 3D, AJ. It may be hard to see. If I pull down here, it may be popping up at a strange place, so you may have to look for the little pull-down menu. But there's the 2D. I pull it again, and it gives me 3D. This one you can see it's pretty small because of the resolution of the screen. Here's 3D, here's X, Y, Y, Z, X, Z, 3D. Maybe Gilberto, you could help people find it. So there was a request for repeating for the contours, the ISO lines for chemical fields. Again, we're going to go to settings, preferences, field tab here. Bottom right, it says scalar contours show. Turn it off, just the value. Turn it back on. ISO lines, isosurfaces. You're going to have lots of time to practice. I'm just giving you a few starting points. And of course, it's important to make sure that you've got things running. Yes. On a Mac, you have to use the default setting, not the light or the dark setting. All right. So you can play a little bit here. Here's 2D, no isosurfaces, 2D with isosurfaces, 3D with no isosurfaces, 3D with isosurfaces for chemical field visualization. Yeah, if you hit the letter R while hovering over the plot, it will reset the plots. There's a good question, which is, can you automatically align the 3D windows? I don't think you can. I think you may have to do that by hand. It would be nice if there were actually a setting for controlling the angle of visualization. Okay. If you want more fields to view, you can have as many windows as you want simultaneously. So you could visualize, say, the chemical field in 2D and 3D simultaneously. Yeah, you could definitely write a Fiji plugin. A CompuCell can call and be called as a Python library. So anything that is Python wrapped, you can call from CompuCell. All right. So I want to move on to just building a simple simulation with Twedit now. And it would be nice if we had more simulations that you could build without any editing at all. And if somebody wanted to help us improve Twedit, we would love to have your help. But for the moment, we'll build the one simulation that we can do with no editing of the code, which is a cell growth simulation. So the first thing we're going to do is launch Twedit. And you can launch Twedit in two ways. If you're in Player, you can hit the little pen and paper icon, which starts Twedit. You could go to File, pull down and say Start Twedit. Or you could go to your taskbar and search Twedit and start Twedit. Your Twedit window, your basic Twedit window will look like this. We're now going to use Wizard. And we could do this in a couple of ways. We could go to CC3D project and then select new CC3D project. That's probably the easiest thing to do. And that will pop up a window that'll say CC3D Simulation Wizard. And you're going to give the simulation a name. You could call it something like Cell Growth Demo. And make sure that the simulation directory selected is someplace you could write to. Okay. I'll walk you through the steps and then I'll do it on screen. Once you've created your simulation, you'll give it a name. You'll select Next. The first window will be general simulation properties. The size of the simulation at X, Y, and Z, periodic or non-periodic boundary conditions, lattice type, fluctuation amplitude, pixel copy range, duration of simulation, initial cell layout, square or round, custom. Units. Typically, one Monte Carlo step will be about 5 minutes. One voxel will be about 2 microns. But we could change those if we want. But for today, we're just going to leave those as defaults. So you don't need to change anything on the screen. You just hit Next. Next, you have to say what your cell types are going to be. You're going to create 2 cell types. You're going to call one cell 1, one cell 2, no space. So you'll type cell type, cell 1, add, cell type, cell 2, add. Then you'll hit Next. Then you'll have the cell properties and behavior. You will select contact, growth, mitosis, volume. You'll hit Next. You're not going to use the chemical field, so you just hit Next. And then it'll say configuration complete, and you hit Finish, and you should get a simulation show. Okay. So let me do that with you. CC3D project, new project. And I'm just going to call this growth simulation. General simulation properties, I don't change anything. Create cell type 1. Create cell type 2. Next. Contact, growth, mitosis, volume local flex. I don't need anything else. Next. No chemical field, so next. No internal contact, next. Configuration complete, finish. Okay. People want to see that again, and people can get through them. You should try to follow along with that. Okay. Step 1, CC3D project, new CC3D project. Everybody got that? Give it a name. Here it's called new simulation. Doesn't matter. And hit next. It'll say general simulation properties. I'm not going to change anything. And I hit next. I now have to create my cell types and call cell 1, cell 2. Be sensible about your cell names. Don't use numbers or special characters at the beginning of a name. Don't put spaces in them, so on. Cell 1, cell 2. Everybody got that far? If you need help or time, let me know. Is it okay to move on? Next. Okay. Under adhesion, you select contact. Under growth, you select growth. Under mitosis, you select mitosis. We're going to be building a simulation of growing and dividing cells. And then under volume, you hit volume local flex. Contact, growth, mitosis, volume local flex. Okay, next. Skip, next, next, finish. So you now are going to go in and double click on the name of the simulation you've just created, and that will open the code for the simulation on the right. I can save the simulation by going to CC3D project, save CC3D project, or if I'm going to give it a new name, which is always a good idea, save CC3D project as. I could also just hit the down arrow. That will save it. I would strongly advise against doing the saves that are not the save everything. The double floppy disk icon saves everything. The single floppy disk icon only saves some things. I would not use that. The one with the pencil lets you rename. Okay. And now we're going to run the simulation. There are a couple of ways we could do that. If player is not running, we can hit the CC3D icon here. We can also pull down CC3D project and say open in player. And we can also right-click on the name of the simulation on the left. It'll pull down a menu and say open in player. Since we had that other simulation running, it should pop up a window saying, current simulation is running. Do you want to stop it and run the new simulation? Hit yes. Okay. So try that. If we've saved the simulation, we could also open a simulation with the up arrow or select File, Open Simulation, and then run. Again, you can manipulate player the way we did before. Start, step, stop. And you should have gotten the simulation. So let's see what happens. Did that work for people? So what did we do here? Let me come back. So Maisha, let me show you here. So here I've created the simulation, growth simulation. I'm now going to right click and say open in player. I probably have to change my screen share here. Screen 2, there we go. I'm going to say open in player. And it says current simulation is running. Do you want to stop and run in simulation? So I say yes. And now I have my new simulation. And this time I'd have to resize the screen. So let me stop this step. That's my initial configuration. And now I can step the simulation. Let me walk you through the steps one more time. I'm going to go to CC3D project, new CC3D project. I'll give it a new name here just for the moment. Create the project, general simulation properties left alone. Add 2 cell types, cell 1, cell 2. Next. Contact, growth, mitosis, volume. Next. 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 Finish. Simulation is up here. Double click on it to view it. Right click, open in player. I have to switch over to player. It says stop current simulation, run new simulation. Hit yes. Now I hit run. And it'll launch the simulation. The window is small. I have to open it up. Let me start it over again. That's my initial configuration. If I don't like the cell color, I can go here to preferences, cells colors, blue, I want it to be purple, purple. Green, I want it to be yellow, yellow. Now we hit run. You'll see the simulated cells will grow and divide. So what we're seeing is individual cells grow and then they divide. If you try to write your simulation, to save your simulation to a directory where you do not have write privileges, then player will crash, Twedit will crash. And so you have to be pointing to a directory that you can write to. So your desktop is not a bad choice. CC3D project, new CC3D project. I can do browse and I could select my desktop here, for example. Anywhere where I know I can write. Next, next. Cell 1, cell 2. Next, cell properties. Adhesion, contact. Growth. Mitosis, volume, local flex. Those 4 buttons. Contact, growth, mitosis, volume, local flex. Next. Okay. Then to run it, select here, right click, open in player. If you hit the circle rather than play, it will display the first condition and then stop. All right. So if you run this, you'll see that it will fill the screen with cells. Let's look at what it's inside of it very quickly, and you'll spend the rest of the day going back and doing this again. We have in CompuCell, simulations are specified in CC3D ML, which specifies static properties of simulation, like what the cell types are, and in Python, which specifies the dynamic properties of simulation. And these are formerly Python classes. There's going to be a certain amount of boilerplate in the Python that you don't need to pay attention to at the moment. If we look at the simulation specification, we will see that there is something called main Python script, which we almost never touch, XML script, and Python. We'll look at the XML first. So we'll look at the XML here together, XML. We have CompuCell revision such and such, version such and such. Metadata. CompuCell is intrinsically parallel. You can tell it how many processors to use. We're not going to change that at the moment. Unit conversion. We're not going to play with that. POTS, the size of the lattice. Number of steps the simulation runs. Fluctuation amplitude with the weird name of temperature. Neighbor order, we're not going to play with. Cell type, we have medium cell type always for empty space. Cell 1, cell 2. Cells have a volume. We're not going to use it, but cells have a center of mass and a boundary. Cells have a contact energy with medium and with each other. And we're going to start out with the uniform initializer that draws a square of cells. And the cells are of type 1 and of type 2. So here are the cell types. Here are the adhesion energies. The initializer sets the initial condition. As I mentioned, we have two cell types, cell 1 and cell 2. These are called plugins. Plugins specify the behaviors of the cell. There's a question, can we add new cells here by changing the XML code? Do you mean new cell types or new individual cells? Yes, we can define a new cell type. If we wanted to add cell type 3, we can do that here by editing the code. So here, cell type 0 is medium, 1, 2, those will display in player this way. I think in the latest version of CompuCell, you can actually specify the color of the cell, but I don't know how to do that yet. Okay. The contact energy defines how cells adhere to each other. You're going to hear about that in the next lecture. Here's the contact energy between medium and cell 1, medium and cell 2, cell 1 and cell 2, cell 1 and cell 2, 1 and 2, 1 and 1, 2 and 2. Here they're all set to be constant, equal to 10, and those will actually show up in player. You can see them there. Uniform initializer draws a square or rectangle. Bottom left-hand corner, box min, bottom top right-hand corner, box max. Width says draw a 7 by 7 cell. The cell types that I'm going to do will be randomly distributed if I list more than one cell type. Gap would give a space between the cells. So why don't you try that in your code? Change box min and box max in your code to draw a square initial cells. Why don't you try drawing just one cell at the beginning of the simulation instead of a square of them? See if you can figure out how to do that. Here's uniform initializer. Box min. I'll try it at 100, 100. Box max, maybe 107, 107. We'll see. Save. If you want me to zoom in here, I can do that, make the font bigger. Yes, you can certainly imitate cell culture experiments. We do that a lot. So now I've changed my initial condition, hit save. And now if I go to player, it should automatically update to the new simulation. Let's see if it does it. Yep, see, I started with one cell now. Everybody able to get one cell to start with? Now you'll see the cell shrinks a little bit and then starts growing and divided. Notice that when it divides, it creates one cell of its old type and one cell of the new type. How could you make it slow down? Well, that's how fast CompuCell runs. But you can definitely make the amount the cells grow per time step smaller. And I want to show you that before we break. So I want to show you the rest of the code now. Again, if people need help, please reach out to the teaching assistants. Okay. Now let's look at the Python. You can go to Twedit, double click on the Python steppables. Why they're called steppables is a long story, but that's what they're called. CC3D specifications in Python live inside of classes. You don't really have to worry about that for the most part. In the Python, you will see a start function, def start. That's where you do your initialization. And there's a command for cell and self.cell list that does something to every cell. You set the target volume of the cell to 25 units. And the lambda volume is how the inverse of the compressibility of the cell, that's set to 2. If I met target volume bigger, the cell would be bigger, grow bigger. If I make lambda volume smaller, the cell is more compressible. If I make it bigger, it's stiffer. Then there is a step function. And in the step function, it says for cell in self.cell list, cell.target plus equals 1, cell.target volume plus equals 1, that increments the cell volume. So the question is, if you want things to grow slower, replace plus equals 1 with plus equals 0.1 or 0.05. There's a question, can you simulate brain tumors? Yes, you can. There's a question, can you simulate proteins inside or on the surface of cells? In general, CompuCell is not ideal for that, but yes, you can do it. And Julio will talk about how to do that later in the week. Okay. The last thing that we have to do in our simulation is have our simulation cells divide. We create a list, Python list, cells to divide. We have to do that because we're not allowed to change the content of a list while we're iterating over it. We go through all of our cells. If the cell has a volume bigger than 50, we say to divide that cell. We append that cell to the list to divide. Then we go through all the cells to divide, and we use the divide cell function to divide the cell. In this case, we do random orientation, but we have options of orienting and dividing in a particular axis, major axis, or minor axis. There's a question, can you save the coordinates of the cells at each simulation step? Absolutely. And Julio will show you actually how to model chemicals that move around on the cell membrane or within the cell. You don't have as much detail as you get with molecular dynamics, but you can definitely do it. There's a question, can you track a single cell throughout the simulation? Absolutely. Every cell has a unique identity. Great questions. Okay. When cells divide, there's a function that says called update attributes that tells you what to do. Typically, all of the parent cell attributes are copied to the child using the clone parent to child, but we typically want to assign half of the volume to the parent and half to the child. So here, line 59, self.parent underscore cell dot target volume divide equals 2 assigns half of the volume to cell one and half of the volume to the parent and half to the child. Clone copies all of the attributes of the cell, whatever they are. That's a very powerful function. And then if the parent cell type was 1, we make the cell type daughter cell type 2. And if the parent cell type 2 was, we make it 1. So if you're editing, there's a question about how to use the editor. Here's the simulation Python. Here's the start function. Here's the growth function. Here's the cell division. Cells to divide. Check if the cells are bigger than 50. If they are, add to the list to divide. Go through the division. On division, update attributes. Divide the target volume. Clone everything else. Change the type from 1 to 2 and 2 to 1. So suppose I make the initial target volume of the cells 49 instead of 25. I hit the save all here, and that will update the code. So save is this double floppy disk icon here, save all. Or I can go to CC3D project and hit save CC3D project. Or, as you point out, you can hit save here. 3 options. Somebody says it goes too fast. I agree, the simulation runs awfully fast. So let me hit stop. Okay, here's my simulation with 1 cell. And to make it easier to see, instead of hitting run, I'm going to hit step, 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 step. Okay. Another way I could do that would be to go into my simulation. Instead of having it grow by 1 per Monte Carlo step, I'll go 1/10 as fast, 0.1. Growth rate. Save. Go back here, stop, step. And now let's run it. It grows slower. You'll notice the cell borders are turned off here. If I want the cell borders turned on, I go under visualization, cell borders, and now it'll show the boundaries of the cells. Okay. Again, we're going to try everything multiple times. One thing that you're going to face a little bit with CompuCell is that CompuCell simulations are not single files, they're a folder. And so typically to download them, they have to be zipped. Twedit will and Player will both automatically unzip CompuCell file archive. This issue with Macs, you may or may not have a problem with at the moment. But when we're going to share files with you, you'll have them in a zipped form. And when you open them, it'll ask you if you want them unzipped. All right, that's all I had for this module. Again, I want people to remember that you're going to have a chance to practice all of these things. This is just a quick overview of all of the different things we could do. There's a comment that Player won't open from inside of Twedit. It's possible on a Mac or Linux installation that that's an issue, in which case open Player separately, and then you can use the open file options, open project options in Player. Thank you to Lorenzo and Priyam and Hayden for answering all the great questions in the chat.